Greetings. Hello, my name is Amberly Lappett. I am a psych major at UC San Diego, specifying in cognitive and behavioral neuroscience. And I'm just recording myself to solidify some information, consolidate these memories so that I can better retrieve them later. But if you get a little bit of information out of this, that's wonderful. This course that I took during the fall 2024 quarter was a wonderful, wonderful course called Cognitive Neuroscience, Psych 108 at UC San Diego, if you're interested. Now, this material comes from midterm two, but it is absolutely relevant for our final, which is why I'm going over it right now. So the things that we went over in these learning objectives was the seeing brain, the hearing brain, the acting brain, and I want to say a little bit of the being brain, but I think that it was just seeing, hearing, and acting brain in this one. So let's get started. We had some learning objectives that we began with, and that's going to be in green, and then any of the answers to it are going to be in red in this part of the seeing brain. Once we get into the hearing brain, the colors are going to shift. So how does information from light make it into the brain? Well, researchers have studied this and they have actually found that there are 10 different pathways that have been actually discovered and then recorded with lots and lots of data that proves and shows these pathways and what their functions are. One very well-known visual pathway is called the geniculostriate pathway, which is the one that we studied specifically in this course. So when light makes it from the eye to the brain, what's happening is photons, or a measurable source of light, will actually come through the visionary field from the right eye and the left eye if you're looking in the right visual field. And if you're looking in the left visual field, information is still coming in from both eyes. But what's happening is information comes through. Let's say that I'm looking to my right. If I'm looking to my right, then there anatomically on your eye, you have, here's your eye. And let's say that this part is the part that is innermost to my nose, right? And this part is outermost to my temple. Well, then this part is going to be the nasal portion of my eye, and this part is going to be the temporal portion of my eye. Okay, and so they used that to be able to talk about terms of reference. So when they're talking about terms of reference, they want to know anatomically what we're talking about. Is it superior? Is it inferior? Is it anterior? Is it posterior? Is it dorsal? Is it ventral? Is it caudal? Is it rostral? Those kinds of questions we want to know, right? Is it lateral? Is it medial? In this case, is it nasal or temporal, right? So with the eyeball, we're saying that information in the right visual field is going to bring in light from the nasal portion of the right eye and the temporal portion of the left eye. But when that happens, when light comes through from the right visual field, on the right nasal portion of the eye, it's actually crossing over the what's called the optic chiasm, which is a decussation, which is a crossover of information, of nerve sensory information. And when it does, it actually makes it to the other side, contralaterally, to the visual field on the other side of the brain. So you would think that when you're looking to your right, that information would be processed on the right side of the brain. On the contrary, When you're looking to the right visual field's information is actually being processed on the left side of the brain. And vice versa, when you're looking to your left nasal portion of your the nasal portion of your left eye and the temporal portion of your right eye are being recruited with neurons that are receptive to that field. The nasal portion of the information is decussating over the optic chiasm and it's coming over onto the right lateral geniculate nucleus of the thalamus. And the temporal portion of the right eye is taking in information and it is transferring that ipsilaterally to the right side also. So both information from both eyes is making it over to the right side of the brain. It's just that the right eye's information from the left visual field is coming over ipsilaterally, meaning on the same side. But information from the left eye in the left visual field is coming over contralaterally to the right side. So that's really a neat phenomenon that we have this contralateral processing that's happening within the visual field within our brains. So it's not to say that when we look to the left, that only the left eye is processing information and then crossing it over to the right, and that when we're looking to the right, that the right eye is the only one that's getting information and then crossing it over to the left. On the contrary, both eyes are receiving information from the receptive field, and it's just that when you look over to the left, the right eye is the one that's moving ipsilaterally to the right side. The left eye is the one that's moving contralaterally to the right side. And when you're looking to the right visual field, your right eye is going to direct contralaterally, and your left eye is going to direct ipsilaterally to the left side of the brain. And so now here's the pathway. So when you're talking about information, light, photons that are coming in through the visual field, more than just what I just explained, there's even more deep processing, or not just deep processing, but this like actual superficial processing that's happening before that light information makes it to the brain. It also has to hit the eyeballs, and the eyeballs have different photoreceptors on them. So the photons come through the visual field and they hit the retina. So the retina is an area at the back of the eye that harbors your photoreceptors. And not only does it harbor the photoreceptors, it actually harbors more like neurons, more cells. They're called bipolar neurons. And then those bipolar neurons are then also um, uh, supported by amacrine cells and horizontal cells. But we didn't go into that in this particular course. But the information gets transduced from the retina, and then the photons will become like a neural impulse, and that neural impulse activates the retinal bipolar cells, and those then send the signal to the retinal ganglia. Retinal ganglia, the bipolar cells then kind of come together and form like a bundle. That's called nerve ganglia. And that bundle ends up becoming our second cranial nerve, the optic nerve. And so on both sides, we have the optic nerve that's coming through. Now, the optic nerve will send the signal through, then decussate over. So information coming through, hitting the retina, activating bipolar cells, activating retinal ganglia. Ganglia turns into optic nerve then decussates, crosses over the optic chiasm, and then the geniculostriate pathway begins, where it's coming across, information is coming across, and it's hitting now the lateral geniculate nucleus of the thalamus on both sides. From there, the information of, well, let's talk a little bit more. So photons are light, right? But then what are the rods and the cones doing? What are the photoreceptors doing in the retina? Like, what is their responsibility? Their responsibility is for picking up light, color, shadows, all of those things. So like when the light is on, the cones are active and we're able to really see all these different colors. When the light is off and when it's dark, our rods are super active and we're able to see different shadows to try and navigate through darkness. Not to say that that the rods aren't active when it's daylight out, because rods are also helping us with like what's called kind of lateral inhibition. Actually, the bipolar cells are really doing that. But being able to differentiate between different shades of shadow. So that is still, I think, necessary in the light. It's not just in the dark that those would be active. So then the bipolar cells, the bipolar neurons, are there to send a signal um, to the retinal ganglia, but more than that, they're also there um, to undergo lateral inhibition, which helps to delineate and see the edge of a shape or an object or an orientation of some kind. So I'm thinking of like letters, for instance, because this was really heavily studied, was like, how do we perceive objects, right? And the idea of perceiving an object takes shape in a lot of different ways. Because let's say we're just looking at these words that are on the screen right now. The H um, of the word how, it has this line that goes down. Well, it has parallel lines that go down, but then it also has a horizontal line that goes across that's a little bit thinner. Well, we have neurons that actually code for each little orientation of that H, each little orientation of this O on the left side of it, on the right side of it, up on the up side of it, and the down side of it. How it, like each font is situated, we have different nerves that will be active for a specific orientation of that edge. And edge detection is actually not in the retina at all. Edge detection is found in our primary visual cortex of our occipital lobe. So that's a part of the lateral geniculate, the geniculostriate pathway. So once the the so the rods and the cones are perceiving light, they're perceiving color, then also um, more fading, more shape of some object is being formed from the bipolar neurons. And then information is being passed through the ganglia, which is making up the optic nerve. And the optic nerve is responsible for sending the signal to the lateral geniculate nucleus after it crosses over the optic chiasm. So now the lateral geniculate nucleus is sending information uh, from the eyes to V1. So any of those little um, edges, the dot detectors, the simple cells, the complex cells, and the hypercomplex cells are now going to be recruited after the optic radiations. So from the lateral geniculate nucleus, there's optic radiations, which are tracks that the information travels through until it gets to the V1, which is the primary visual center of the occipital cortex. So and then V2 gets stimulated, where like V1 is responsible for detecting edges with the simple cells and the complex cells and the hypercomplex cells, and they're all doing different things. The simple cells are more responsible for detecting different dots as well as the edges, but then the complex cells will detect the edges but not the individual dots. And then the hypercomplex cells will not only detect one edge, but it'll detect multiple edges to be able to form an actual shape of a letter. So then beyond that, we have even higher order recruited visual areas. So V2 is another visual cortex or area of the visual cortex that helps V1 um, transfer information more strengthened to V3 and V4, which has to do with color perception, but on a deeper level, cortical color perception. And then V5, which has to do with the movement of an object. So not only are you perceiving like the color of the object and its orientation of an actual object. It's not just a blur of color and something there, but now you're actually forming something of what it is. And that's all a part of your V1, V2, V3, V4, V5. But now you're recruiting even higher order areas of your brain, which are called the what, where pathways. So your ventral area of your brain is going to be able to detect what that object is. You know, you can recall it, you've seen it before, you know what it is. Oh, it's a tripod. I know what that is, you know, kind of a thing. Um, and then also where it is. Oh, I see a tripod. It's here and it's sitting in front of me. I know that if I reached out for it, I know where to grab for it because I see where it's at. That's called the dorsal stream of information. That's going through more of the parietal and occipital lobe versus the temporal and occipital lobe. So those are the different areas that you get active when information from light is making it to the brain. But also, I just told you what the geniculostriate pathway was and how information from both visual fields travels down that pathway. So again, the geniculostriate pathway is the primary pathway for the perception of objects and faces from the receptive field. And information from the right visual field travels through the nasal medial region of the right eye and temporal lateral region of the left eye, and vice versa. Information from the left visual field travels through the nasal or medial region of the left eye and the temporal or lateral region of the right eye. And the nasal stream of information from both eyes is crossing over, decussating at the optic chiasm, and each temporal stream travels ipsilaterally down the geniculostriate pathway, and then synapses with the connecting nerves at the lateral geniculate nucleus in the thalamus, the central relay station of the brain. It should really be central sensory relay station of the brain because there's a motor relay station as well, or a motor area of the brain as well. So this is just a central sensory relay station of the brain. And the information travels from the lateral geniculate nucleus to V1 via the optic radiations to the other higher order centers of the brain. So then what types of information are processed in V1 and by what kinds of neurons? And that's what I was talking about next. I kind of gave you an overview of everything that I just got done saying. Where V1 is a primary visual cortex that receives projections from the lateral geniculate nucleus and perceives basic visual information such as multiple dot detections. Remember, I was talking about that with the simple cells. But then it also will be able to see edges that are created, lines that are created, basic orientations and directions from like the simple, complex, and hypercomplex cells that I was saying. And that's going to send other more complex visual information and the processing of that information to secondary cortical areas. And those are the V2, V3, V4, V5, and even higher order, the what, where pathways. So the information derived in V1 forms the building blocks for more advanced responses of neurons higher up in the hierarchy. That's the what, where pathways, the dorsal and ventral streams. So what are the secondary visual areas and two higher order pathways, and what are their roles in vision? So here's an overview where V2 helps V1, like I was saying, distribute visual stimuli to proper brain regions for visual processing. V1's assistant kind of thing, where it helps process and communicate basic information from V1 to other brain regions. V3 is super complex. There are a lot of different things that V3 does, which is why we did not go over it in this course. But V4 is all about the perception of color. V4 tries to compute the color of the object by taking into account variations in lighting conditions. So where the rods and the cones are kind of doing that too, now you're looking at the color of an object and the variations of lighting there. So like sometimes it looks like an object is kind of gold and white when the light looks like it's coming off of that actual object versus in the background. Where if the light was coming from the background or it appeared to come from the background, now that object looks like it's a totally different color, even though it's the same exact object. So those are the kinds of things that V4 will recruit. Um, Cortical color blindness is actually an issue in this V4 area, where it's more of an abnormal color blindness. Your normal, typical color blindness that you hear about is like where you can only kind of see like red and green tones, but you can't see any other tones. And that's from the retina, and that's because like you don't have enough cone cells. But cortical color blindness is where V4 gets damaged, and there is a phenomenon called achromatopsia, where every color is gone and everything appears in grayscale then. So that's an interesting phenomena where you just have a failure to perceive color at all. But it makes everything like this grayscale. It has nothing to do with retinal cone receptor damage. The retina are still functioning normally in this case. So then V5 is perceiving an object in motion. And all of that, all of those different regions of visual pathway is evidence for localization of function, meaning that we have different structural functionalization. We have different structures, different cortices of the brain that are responsible for a specific function. And so now we've been able to localize a specific area on that cortex of the association area that has to do with a specific task. So in this case, vision, right? And we were able to find that V1 does something completely different than V2, than V3, than V4, than V5. But there's such thing as the binding problem, which is where we still don't know how these areas of the brain, these regions, these association areas are speaking to one another. So that is still an area of research if you're interested. Now the ventral what pathway is one of the higher order pathways of vision, and it's responsible for, like I was saying, recognizing form representation. So it's looking at like how something is oriented in time and space. Is it sideways? Is it backwards? Is it frontwards? Is it upside down? You know, those kinds of things are going to be activating the ventral what pathway of the parietal, or I'm sorry, of the temporal lobe. Form representation, including face recognition. So like your configuration, I'm sorry, like that was totally messed up. Strike that, reverse it, and let's go again. The ventral what pathway is responsible for recognition, so recalling something and knowing that you know what that thing is, like knowing that this is a tripod, right? That is recognition. I recognize what this thing is. I recognize that this is a bolster. I recognize that these are flashcards, right? Those kinds of things. That's recognition. That's a ventral pathway that's in the temporal lobe. temporal occipital lobe, and face recognition is also there. Now the dorsal pathway is going to be along the parietal and occipital lobe, and that's the where-how pathway that's responsible for the guidance of actions and where something is in space. That was what I don't know why I said the other way around. But yeah, so where something is in space is your dorsal pathway and how it's oriented in space. So when I was talking about before, if it's facing forward, if it's facing backward, if it's facing on its side or diagonally or upside down, that's all a part of the where-how pathway that's going to be active when you're thinking about those things. So it's not to say that the ventral what pathway and the dorsal where pathway are lit up at different times. They can definitely, it's a parallel processing to kind of an activation. So that is what I also see, even with this binding problem issue, is that really it's just it's being parallelly processed. In other words, one area of the brain is activated at the same time another area of the brain is activated, even though they are doing two separate things. So these are the higher executive pathways, or streams for retrieving like top-down information. In other words, general information that you've gathered from your experience in the real world and being able to then kind of excise that down and dissect it into smaller parts to be able to figure out what it is. So familiar objects, faces, words, those kinds of things are going to be easier to decipher through and dissect down through a top-down process, where you're able to recognize names and, you know, recall memories, different episodes from your past. All of that's going to be like this occipital temporal ventral stream of information that gets activated. Temporal occipital ventral stream that's going to get activated to recognize things, objects, faces, names, putting memories to things. That's all temporal lobe. And then where you see the object in space so that you can pick it up without missing it. That's all the parietal occipital dorsal stream or occipital parietal dorsal stream. So now what types of blindness does damage to the retinotopic map? Well, what is retinotopic map? What is that? Well, it has to do with what they've been able to see that the intelligence that consciousness does for itself or genes do for itself, they are able to form a retinotopic organization where um, information and what objects that are being perceived from your visual field or your receptive field are also like things that are close together out there are going to be like activating different neurons that are close together inside the brain. So that's kind of the retinotopic information. It's saying that in the retina, there's going to be cells that are close together to be able to transduce and send a neural signal to the bipolar cells that say, I'm seeing something and this is what it is, then the signal, so that is retinotopic organization. It's saying that something out there close together is also going to be recruited close together with nerves in your retina. So what do I have here? Cortical blindness from V1 is what type of blindness that does damage to the retinotopic map. So why? Because the information and the mapping that's happening from the retina is actually being transferred to V1. So the retina is still functioning normally. It's that damage to parts of area V1 are going to result in blindness for the corresponding region of space because of the retinotopic map. So the information in the retina gets transferred back, crosses over the optic chiasm, comes to the lateral geniculate nucleus, and then goes back to V1. So all of it's getting projected. And anything in V1 is seeing what's in the retina. So if the retina are intact, then it has to be V1 that is that's causing the damage, the blindness, uh, to the retinotopic map. V4 would be cortical color blindness. What are the four general types of object recognition? So to provide examples and evidence of each of these, we saw this. We saw that there was early visual processing that takes place. And during the early visual processing, that's like when the light is coming into the eyes, through the retina, through the bipolar cells, through the ganglion cells, through the optic nerves, past the optic chiasm, into the lateral geniculate nucleus, and into V1. That and V2 and V3 and V4 and V5. All of those things are early visual processing. So that whole thing is going to help us perceive color, perceive motion, perceive edges, and the depth, all of it. Now, when we're able to group visual elements into some sort of a mental representation, that's going to recruit the ventral wetware pathways. That's a part of the Gestalt principles of grouping. So like the law of proximity and the law of closure and the law of good continuation, and the law of like grouping. These are all found from the Gestalt principles, saying that we tend to, when we see something that like are the same colors, and let's say they're all the same colors going down like this, and then you have another row of a different color going like this, another row that is the same color as the row before that, you're seeing that those are columns. In your mind, you're perceiving a pattern saying, oh yeah, those are columns. If you see groups of dots over here, and you see groups of dots over here, and you see groups of dots over here, and you see groups of dots over here, well, you're going to group those together because they're close together. That's the law of proximity. If you see, let's say, a rectangle here, but then in front of the rectangle, you have like a cylinder type of a shape, right? If these lines match up behind the cylinder, well, that is the law of good continuation, saying that it looks like there is a rectangle that is all together like a brick behind this cylinder type of a shape. So it's saying that even though there's something in front of it, your mind is perceiving that there's something together there. So those are the types of ideas of the Gestalt principles, of grouping visual elements into mental representations. There's also Gian grouping, and Gian grouping is like the three-dimensional grouping versus two-dimensional grouping the Gestalt principle does. Figure ground segmentation is another one, but we didn't really go over that much. Now, matching the mental representation to knowledge in the brain. Once we have grouped those visual elements into a mental representation, now we have to like match whatever that object is or that face is that we see to something that we've seen before to know what it is, right? So that is that mental representation to knowledge in the brain, the semantics or episodic memory, declarative memory, so that now we can say, yeah, I've seen it before, or no, I have never seen that before in my life. Then comes the naming part. Have you seen this before? Yeah, I've seen it before. What is it? That's Mary, or that's my old house, kind of a thing, right? So that is attaching a meaning and retrieving that semantic memory. right? So that's all going to be from the temporal lobe. Now, what is the role of the LOC in shape perception and object constancy? Well, we didn't really go over that um, in the first part of it. So unless you're familiar with the the LOC, it's like, what is that, right? So it's called the lateral occipital cortex. And the lateral occipital cortex is found in this ventral what pathway, like in the kind of more posterior, closer to the occipital lobe, kind of like the junction between the temporal lobe and the occipital lobe. Um, and it's helping to identify objects even when the object is under special conditions, like a change in the size or the change in the shape of the object. So they were able to do bold testing. Bold testing is a type of neuroimaging study with functional magnetic resonance imagery, and it's where you're utilizing the biology of the body and what it wants to do when a certain tissue needs to get activated. So when innervation needs to take place, when tissue needs to have needs to give rise, blood needs to be recruited to that area so that oxygen can go to that area to nourish um, for cellular respiration so that you can get energy to the area so that it can do work. So, um, all that to say that for the lateral occipital cortex and like shape constancy, they did a test with a bold signal, the blood oxygen level dependency. So what that's doing is it's measuring with an fMRI when they give a stimulus and then you have to respond. Well, you're like having to and when you see something, that's like kind of the response, like a yes or no kind of a thing. Did I see it? Did I not? Um, that's activating a certain part of your brain. Well, if you've already seen it before, then your brain's already been primed for it. And um, what evidence has shown is that the activation of those nerves that are firing to recognize that object and to see that, where it is and everything, um, those fire less if you've already seen it. And there's a phenomenon called shape constancy and size constancy in objects. And the LOC, or lateral occipital cortex, is involved in perceiving these different shapes in objects. Um, so what it will do is it will fire less if it's seen the object more than once, even if the object is the same object, if it's the same object but a smaller size of the object. But if the object is oriented in a different way, like let's say it's facing forward this way, but then it's facing sideways, that area of the brain, lateral occipital cortex, is not activated when seeing it twice. Like it's okay, it's activated, but it doesn't change. It doesn't lower the activation um, the second time. In other words, that area of the brain has not been primed for that, which means that is not the area responsible for orientation constancy. It's for shape constancy and it's for, or I mean, object constancy as well as size constancy. Because they were able to see that if you had like a larger umbrella size and you showed them that image, the LOC lit up. Then you showed them the same umbrella but smaller. The LOC lit up but a lot smaller as far as the activation went. So that gave um, evidence that the lateral occipital cortex is responsible for shape perception and object constancy. So what did I say here? We still know and can recognize the object, measured and confirmed recognizable with the LOC by measuring bulb signals when shown certain objects in different orientations. LOC responds more to viewing objects than to viewing textures, but responds the same to real and made-up objects. So the TMS, that's transcranial magnetic stimulation. It's a type of neuroimaging that you can do where it's real-time manipulating brain activity to see if it's necessary for a certain function. So they did that to the lateral occipital cortex, and they were seeing that it actually disrupts your ability to match objects by shape, but not their orientation. So the LOC is not responsible for detecting orientation, but it is responsible for detecting shape and size. So what does it mean if there's a significant difference in bulb signal between 1 and 2, meaning trial 1 and trial 2, for a particular brain region? It's saying that the response of neurons tends to be lower for repeated stimuli. This is termed adaptation or repetition suppression. So that's the phenomenon. And we've measured that with the bold response in the fMRI. So how does face recognition relate to object recognition? Well, this is actually really fascinating because they were able to see that face recognition generally mimics the processes of object recognition. But there's a difference in the end. So like you're still going to structurally encode for basic visual features of like the configuration. That's the early visual processing part, but you're structurally encoding the face. Then you are grouping the elements into a mental representation of a face. And then you match that mental representation to your knowledge bank of if you've seen that before, that face before. And then, oh yeah, I've seen that face before, and you name the person, right? So that's all a part of face recognition, and how it's relating to object recognition is really mimicking the broad stages of object recognition. The difference is that if you have deficits in stages 2, 3, or 4, you're not going to be able to recognize faces at all. That's face paralysis, or what's called prosopagnosia. And that's the inability to recognize familiar faces. So prosopagnosia has definitely been studied, and that was a way that they were able to determine that there is a face area for recognition. They call it the fusiform face area. So they're saying that that prosopagnosia typically results from damage to this fusiform face area or other regions in the ventral wet pathway. So question, do you think faces are just complicated objects, or do you think faces are special? Well, turns out that facial recognition has a very specific neural substrate, the FFA, so that makes it pretty special. Also, nerve receptors respond more to whole faces and the actual configuration of the face. So if it's like upright versus like upside down. So if it's right side up, you can recognize the face better. If it's upside down, it's a lot more difficult to recognize a face. If you were to, on a computer, take a right side up face and take the features of the eyes and the mouth and flip them upside down, it's harder to recognize a person in that way than it is if the orientation is proper. So that's really interesting. Um, so yeah, so the receptors, the nerve receptors for facial recognition are responding more to the whole faces and their configurations versus the primary parts of a face like an object does. So that is another part that's different between the face and the object. where objects get recognized more with this bottom-up processing, and faces get recognized more with a top-down processing that rely on like a holistic versus part-based processing, the face. And um, faces are special because you have to have some sort of a level of expertise to discriminate between faces. So, also, faces are a distinct category compared to objects when it comes to, you know, how they are recognized. And we were able to do double dissociation studies because of this fusiform face area to be able to differentiate between how objects are recognized and how faces are recognized. So they did that by studying the parahippocampal place area and the fusiform face area and did double dissociations with people that either had FFA damage or PPA damage, but not the other. And then they had both participants do the same tasks that were predicted to activate those centers of the brain. So they showed them pictures of faces and they showed them pictures of houses. And they were able to see that the people with damage to the parahippocampal place area were unable to recognize that the places that they had seen, the houses that they had seen, but they were still able to recognize the faces that they had seen. And same with the people that had FFA damage, those people were able to recognize the places that they saw or houses, but they were not able to recognize the faces as well. They made a lot more errors. So that's what you're measuring in a study like that is accuracy and looking at how many errors are taking place when somebody is trying to recognize something. And that's how you do the double dissociation study. It was really cool. So let's see how much time that was. I have no idea. I don't know. Anyway, so that was the seeing brain. Now for the hearing brain. What are the physical components of sound and how do we perceive them? The hearing brain is probably one of the coolest parts of the body. Um, I think that all sensory pathways are so amazing, so fantastic, and such a privilege to be able to study on this level because there's so much to be known in this way. So, and yeah, there's just so much to discover because of being able to know about the way that these things work. Um, so what are the physical components of sound? Well, sound is a change literally in the air pressure over time. So as the pressure of air comes through the small tunnel of our ear, it's going to form like a wave, kind of like a sonar wave, like right, like right. But that wave, like a barrel wave, is pressure, right? And so the physical sound waves are consisting of like a frequency, right? Hertz. And that frequency is either going to be strong, and those are the amps, right? The higher the frequency, the stronger the sound, the louder it is, right? You're perceiving it as a pitch, the frequency, and the loudness or the amplitude. But the physical sound wave is frequency and amplitude. So, not that we hear pure tones in reality, but a pure tone is a single sinusoidal wave. So what does that mean? Well, like think about just the wave that goes up and goes down and goes up and goes down like a cycle. That's a sine wave. So like in calculus, in pre-calculus, trigonometry, they talk about sine, cosine, and tangent. And that's because of the way that curves or lines will go on a graph. And that's because of a specific mathematical equation due to the phenomena of nature. And so they were able to see in the phenomena of the nature of sound that there are sinusoidal waves that make up pure tones of sound. And it's beautiful, but it's rare that you ever hear just one pure tone. You're usually hearing complex amalgamation of several sounds that are going on in the atmosphere. And so it's like multiple sinusoids that are together, and either they're going to be um, convergent or divergent. In other words, they're either going to come together nicely or they're going to have dissonance to them. So it's either resonant, where it all sounds beautiful and perpetuates, or dissonant, where it ends up crossing over the sound and cancels it out. So um, the superimposed pure tone sinusoids are different frequencies, intensities, and phases, where frequency relates to pitch, like I was saying, amplitude relates to loudness. Pitch and loudness are the psychological properties, where frequency and amplitude are the physical properties. So note these two types of auditory interference, like I was talking about. So like the dissonant interference would be destructive, and the resonant interference would be constructive. So the constructive interference of audition is where two sound waves are in phase. That means that they're not crossing, they're not touching where the peaks are. They're not like where the trough of one sinusoidal wave is, it's not coming together with the peak of the other auditory wave or sound wave or pure tone. Okay. So that would be constructive when they're not doing that, when it's like this and then like this right underneath it. But destructive would be where they're out of phase. Okay. So then perceiving pitch is where like your musical notes typically contain, a series of regularly spaced sinusoids. And when they're regularly spaced, they're multiples of each other. And so in that way, like think of the way that a piano note goes, like do, re, mi, fa, sol, la, ti, do, ah. Ah, ah, ah. Those are all multiples of one another. So if you have an A sound, that's 220 hertz. You can multiply that frequency by doubling it and make it 440 hertz. And you multiply that by doubling it, and it gets to 660 hertz, and so on and so on and so on. So that is multiple sinusoids together. And that's how we are able to perceive a pitch, when there's multiple of those that are coming together. Now, the lowest component of sound is termed the fundamental frequency. So F naught is going to be that fundamental frequency that is perceived by the brain. So it's really interesting the way that this works, though, because we have evidence of pitch constancy in that there's this missing fundamental phenomenon. It's a trip because when F naught is missing, in other words, if you had a piano singing 220 hertz, dum, and then you had another one at 440 hertz, dum, and then you had another one at 660 hertz, dum, and you played all of them together, dum. If you take away this, you still perceive that in your mind as the so the lowest frequency is the one that's perceived in the mind. The missing fundamental phenomenon is saying that even when you take that F naught away, that the fundamental frequency, um, it's still the pitch that's perceived in the mind. An example would be like two notes with completely different physical characteristics, yet they still have the same pitch to them. How does that make sense, right? I'm still trying to wrap my head around it. Now, how does the neural signal for sound travel from the ear, the inner ear, to the auditory cortex? Well, it's kind of nice that they tell you from the inner ear to the auditory cortex because there's an outer ear, a middle ear, and an inner ear. Okay, so the outer ear is a part of the external um, auricle, and then from there you've got your um, external acoustic meatus, and that's like the acoustic canal, which is like the tunnel that goes to your tympanic membrane or your eardrum. All of that is a part of your outer ear. Once you get past the tympanic main or tympanic membrane or your eardrum, now you're going into the middle ear. And from the middle ear, that's where like all of the small ossicles or bones, or they're called like auditory ossicles. So your your malleus, your incus, and your stapes. Those are the three bones, and they end up getting the physical vibratory signal that's coming from the outer source, where the waves are coming in and pushing on that tympanic membrane that then pushes on these bones that then push on the oval window of the cochlea. So now the cochlea is a fascinating part of the ear where uh, it converts liquid-borne sound into neural impulses. It's fascinating, absolutely fascinating. Um, but now those neural impulses are acting on what's called the basilar membrane of the cochlea. So that's where the membrane within the cochlea contains these tiny hair cells that are linked to neural receptors that are making up the harmonics of the sounds that you know in audition. So it's like the home of the first tonotopic organization for sensing pure tones. Tonotopic organizations like retinotopic organization just for sound waves. So the physical sound waves that are out in the outer world are being perceived in the inner world. And the first tonotopic organization for those sounds are coming from the cochlea in the basilar membrane. And then they go back to the auditory pathway, or auditory cortex, should I say. So now sound induces medical or mechanical movement of the basilar membrane, which initiates neural activity in the auditory nerve. So the auditory nerve in this case is the cochlear nerve. So it's called the vestibulocochlear nerve, which is the eighth cranial nerve. But there's two different branches, the vestibular nerve and the cochlear nerve. So the vestibule has to do with like balancing in the semicircular canals. So like being able to keep yourself oriented and being able to do this without like spinning and falling sideways. That's all a part of the vestibule or the vestibular nerve. Now the cochlear nerve is a part of the cochlea which has to do with the audition part, and that's going back to the primary auditory cortex, A1. So sound induces mechanical movement, initiates neural activity in the auditory nerve, and then the location on the basilar membrane that vibrates most strongly is what's going to determine the perceived pitch of the sound. So so the cochlea is like a snail type of a structure, and within that whole structure, sound is coming through and vibrating. And so depending on where it is, if it's at the apex of the cochlea or if it's at the base of the cochlea, it's going to have a different pitch. So towards the base of the cochlea, you're going to get a higher pitch. Towards the apex of the cochlea, you'll get a lower pitch. So now from the auditory nerve, the signal is traveling to the brainstem, Okay, so now this has gone from the physical mechanisms through the tympanic membrane, through the ossicles, the middle ear. Now we've moved on to the inner ear, which is the cochlea, and the basilar membrane is like saying, okay, that's the perceived pitch of sound, and now that's taking it from the cochlea and sending the signal through the nerve, the auditory nerve or cochlear nerve, right? And that nerve is traveling to the brainstem. So the brainstem's taking on the information, and specifically between the pons and medulla, there's a junction, and the pons medulla junction has this specific region where there's a receptor for audition. So the auditory nerve will travel and synapse there at the brainstem, and then another nerve travels to the midbrain, um, um, where there's another synapse that takes place, and then from that place, another nerve travels to the medial geniculate nucleus of the thalamus, and from the medial geniculate nucleus of the thalamus, information gets transferred to the primary auditory cortex, or the core region of A1, or I mean the core region, which is called A1. And then there's the belt, or the secondary auditory cortex, which is A2. And then the parabelt, which is another secondary auditory cortex. But it's like, if this is the core, then the belt is here, and then the parabelt is around it like that. So it's like core, belt, and then parabelt. Different regions of the auditory cortex. Primary is A1, and the secondary auditory cortices are A2 and A3. So just note that auditory cortex is mostly in the Heschl's gyrus in the temporal lobe, and information received by the core, so A1, is sequentially processed by the belt and then the parabelt. So it's like the core, then the belt, then the parabelt. It's sequential. What are the similarities and differences in visual and auditory systems? So they're really similar because information is being processed from like the early visual centers as well as like early auditory centers, right? What did I say here? So these are the comparisons, and basically they both have a thalamocortical route. One of them is traveling through the lateral geniculate nucleus, which is visual center, but then the other one is traveling through the medial geniculate nucleus, which is the auditory center. Lateral geniculate nucleus projects to the primary visual cortex. Medial geniculate nucleus projects to the primary auditory cortex. And as far as organizing of early neural processing, well, auditory system has tonotopic organization, meaning orderly mapping between sound frequency and position on the cortex. And then the visual system has retinotopic organization, which is also orderly mapping, but between the position on the retina and the position on the cortex. So this is a difference. So those two were similarities. Now, when it comes to differences between audition and visualization or visioning, temporal and spatial sensitivity. So, which one is a faster route? And then which one can be perceived faster? So, audition is going to be more temporal than spatial. In other words, you're going to be able to perceive the sound before you understand what that thing is and where it is. So before you even know where a sound is, you're going to know that the sound is there. When it comes to visual systems, you're going to know that something is there before you pick up on what it is. And you might have a reflex before you actually know what that thing is. If you see it in your vision, right? It's like with audition, if you hear a gunshot, what's the first thing that you do? You duck, right? Or you cover your face or you do one of these things. That's because even though you don't know where it is, you're still going to react. So it's a faster reaction. Whereas your spatial and temporal with visual center, um, if you see something that comes at you, the first thing that you're going to do is jump because you see it right away. It's there. So that's the way that I perceive those two. Um, as far as like functional specialization of feature processing, now these two are also different. So like when you're thinking about like specialized functionings, right? We know that in the visual center, we've got a well documented for color and for movement. V4 and V5 are very spatial. We know for sure that those areas of of the brain are specifically for that spatial resolution, right? Um, it's less well documented for audition for the auditory domain, in that, and that's a part of the temporal also, is that like we hear what's going on and we're able to like see the wave quickly and we're able to see it pick up on neuroimaging quickly. But as far as knowing specifically where and what is happening in that area, it's difficult to say because there's a lot of overlap. So that's another part of what this temporal and spatial sensitivity is all about. Higher order context dependent pathways. So now you can see that when we were talking about the higher order pathways, we were talking about the what-where pathways of the ventral and dorsal streams. So context dependent means that it depends on what type of scenario it is on when these things are going to get active. So there's evidence for separate auditory pathways when it comes to the what versus where and how. There's also evidence for separate visual pathways for what versus where and how. So they're saying that both of these, audition and visioning, have to recruit both both what pathways as well as where pathways, and that those two areas for both are separate, which is really cool. It's saying that, yeah, we do have a what pathway that is totally separate from our where pathway, and both of them have to be recruited for audition and for vision. So what about evidence from single cell recordings? What have we been able to find? Well, that is our evidence for temporal versus spatial, because we can find a lot more spatial information if we can narrow it down to a single cell recording versus having to rely on oscillations and not knowing if we're picking up on more activity than just the stimulus. Just, it's hard to control and manipulate in that way and know that it's objective. So compared to vision, there is less evidence that different auditory features like loudness and pitch and tempo are localized to individual regions. That's what I was trying to say. So they are temporal in nature, just not spatial that we know of. And the strongest evidence points to like a pitch center related to pitch constancy in secondary regions. So remember, the secondary regions were the belt and the parabelt. So they were able to do single cell recordings in that area to find like this pitch center, but that's really hard to do because of the location of audition. So like it's more medial on the temporal lobe, whereas the visual centers are more superficial on the occipital lobe, so they're going to be easier to get to than these auditory areas. Um, so what are the components of the auditory cortex and the dual auditory systems, and what are their functions? So again, primary auditory cortex is A1, and A1 is responsible for detecting pure tones and sound. But the secondary auditory cortex of A3 and A2 are specifically there to pick up on complex sounds and sound patterns, including speech. So the auditory pathway is not a passive transmission of information. Actually, it actively extracts and synthesizes information from the auditory signal. So it's discriminating, and it's also selectively attending to a specific sound wave or specific sound waves, and also where they're coming from. So when you are selectively attending to something, then you're focusing on that thing, and that's what's being picked up on. But the idea of dual auditory streams comes from some auditory neurons that are tuned for the sound itself and then where the sound is coming from. So the sound itself is like, I hear it, but I have no idea where it's coming from, right? And then you know that the sound is over there, but you don't know what you're hearing. You know what I mean? So like let's say um you hear a baby crying. You know that it's a baby crying, but you have no idea where it's coming from. You can't tell if it's coming from over there or if it's coming from over there. That's the sound itself, right? But then where the sound is coming from is like um okay, I know exactly that the sound is coming from right over there. And let's say it's a song, but you can't make out what the song is, but you can hear that there's music coming from over there, that kind of a thing. So and that's the dual auditory stream, where there's two different regions or areas of the brain, association areas, that are tuned for the sound itself and where the sound is coming from. But they're like being parallelly undergoing parallel processing, meaning both of them are active at the same time, even though they're doing totally different, they're doing two different things. So it's again a part of that binding problem thing. It's like, how is it that this region of the brain is talking to this other region of the brain to execute a specific response behavior? So how are sounds located? Well, they were able to see that there was a couple of different ways that you can locate sound. Mechanism one is this idea of interaural differences. So interaural differences is about like the timing difference. So like, and also like the loudness or how intense is the sound. That's going to determine how you can locate a sound, right? So like if it's just a muffled, really low sound, it's really difficult to make it out and to know where it's coming from. But if there's some timing differences and it's not just constant, and if there's some intensity behind it, then you can kind of pick up on where it is. And then also mechanism one is saying that, you know, that this kind of a mechanism, interaural difference, is showing you location for sound on a horizontal plane. So like if you're looking left because you hear a sound coming from over there, or if you're looking right because you hear a sound coming from over there. Okay. So interaural differences, it's like if you hear something over here, it's automatically making your head move over there because you hear it. Or if you hear something over here, it's automatically making your head move over there so that you can hear it. But this part of your nose is interfering with sound. So, an example, a neuron that is selected for a particular frequency may be more responsive, generate more action potentials when the left ear receives a sound slightly before the right ear, but it may reduce its responsiveness if the right ear receives the sound first, right? So it just depends on which ear is picking up the sound first and how much of a hindrance there is because of it, there being over here versus over here. So the second mechanism is distortion by the head and the outer ear. So I guess that's what I was talking about with this planum temporale, um, that compares an internal model of your individual head. So like there's something inside your mind called the planum temporale that compares an internal model of your own individual head and your ear shapes to infer the location of the sound. So if you move your head, it's going to change the way that the sound comes in. And if you move your head and the sound changes, well, then you're going to have to move it again to see where the sound is coming from. So it's important for locating sounds not only in the horizontal plane, but in the vertical plane too. So distortions by head, right? You're moving your head and your ears are moving, the outer ear, and the way that the pressure is coming in is going to be different. What is congenital amusia? And what is the neuroimaging evidence that is associated with it? So what is amusia? Amusia is a scientific word for tone deafness. So like congenital amusia is a developmental difficulty in perceiving pitch. So, like, you can't detect when someone sings out of tune. You can't detect when you sing out of tune. You can't even recognize familiar songs and sing without the help of lyrics because of amusia. Um, you you can maintain songs in memory. Um, or no, you have difficulty recognizing familiar songs. You can't sing without help of lyrics, and you can't maintain songs in your memory. That's all congenital amusia. But congenital is meaning that it's been there since birth. So congenital amusia is that it's emerging since birth, and you have no delays in your speech, you have no intellectual deficiencies, you have no acquired brain damage, and you have no music deprivation. You have a normal understanding of speech and prosody. So, what is this phenomenon about? What is happening? Well, they were able to see that congenital amusia is actually linked to increased gray matter in auditory cortices and frontal regions. So, uh, it's reducing the connectivity where you don't have as much white matter tracts in the right arcuate fasciculus. So you've got more gray matter, but less white matter tracts that are allowing the links to be able to be made for pitch perception. So it's this anomalous recurrent processing where your intact representation of music pitch in auditory cortex is a bottom-up process, and poor feedback control between the right inferior frontal gyrus and auditory cortex is going to be that top-down issue with anomalous recurrent processing. Okay, so the acting brain is the last part. I want to say, yep, hopefully I can get through this in 10 minutes. All right, acting brain. So how does the frontal lobe contribute to movement and action? So the acting brain, when we're talking about acting brain, we're not like talking about somebody who does performing arts. We're talking about any voluntary movement of the body. So voluntary movement has to do with the skeletal muscle tissue, but it's more than that. It's about recruiting the executive function of your frontal lobe. So how does the frontal lobe contribute? Well, there's many areas of the frontal lobe. There's the primary motor cortex, the premotor regions, and within them, there are different subregions. So within the primary motor cortex, that is where voluntary movements are initiated. Now, the premotor regions are involved in this online coordination of movement. So, like, I know that I need to do this thing and I need to do it coordinatingly so that it all kind of works fluid. Otherwise, I'm going to be like clumsy and I'm going to trip over myself, right? So, the premotor regions are involved in the coordination of movement. Now, the prefrontal region are the regions of the frontal lobe having to do with planning and selecting actions according to goals. So planning and selecting actions according to your goals, that's all like here, like prefrontal region is here, premotor region is here, primary motor cortex is got the sensory somatosensory area of the brain. It's like pre-central sulcus, and then like the post-central sulcus or the anterior central sulcus, which is like the that primary motor cortex. So like right in front of the homunculus. Um, that's where voluntary movement's initiated, or the M1 is located. But then within that, you've got these premotor regions, prefrontal regions, etc. So the premotor areas assist with preparing actions. Okay, premotor regions will prepare the actions, online coordination. But now the frontal eye fields are all about the voluntary movement of eyes. So like moving your eyes left and right, bringing them together, like that kind of thing. Lateral premotor cortex links the action with visual objects in the environment, and the medial premotor cortex is the supplementary motor area. Okay, so premotor regions, lateral premotor, medial premotor, and frontal eye fields. Those are the three. So like you see the frontal lobe here, it has so much to it. Like there's a lot of tissue there. You can see that the lateral premotor cortex is the green here, but then the frontal eye field is this lighter blue part, and the darker blue part is the prefrontal cortex. And then the like medium blue is the anterior prefrontal cortex. So this is all prefrontal cortex as well. Anterior prefrontal cortex, dorsolateral prefrontal cortex, front eye field, lateral premotor cortex, primary motor cortex. So the premotor areas, right? that's this lateral premotor cortex thing. And then right, the premotor area, part of that was the frontal eye field. Let's see, premotor areas, frontal eye field, lateral premotor cortex, medial premotor cortex. So here you can see the primary motor cortex a little bit better here, but this is the lateral view, this is the medial view. So this is the right hemisphere of the brain, and it's showing you medially, if this line right here is the central sulcus, this back here would be where the homunculus is on the somatosensory cortex of the parietal lobe. But then this is all frontal lobe here, primary motor cortex, and then supplementary motor areas, right? The medial premotor cortex, lateral premotor cortex, and the SMA, or the supplementary motor area, all right here. And then prefrontal cortex is all right here. So the prefrontal cortex is generally involved with planning and higher order cognition. It's not just action. It's about like your personality, how you set goals, how you maintain your goals. All of that has to do with your prefrontal cortex. So after your premotor cortex prepares an action, right? Let's look back up here for a sec. The premotor cortex, once it prepares an action, it's also going to mediate the selection of the action and maintain the goal of the action. Now, again, the homunculus. The homunculus is a part of M1, the primary motor cortex. The somatotopic organization of the primary motor cortex is saying that there are specific areas of M1 that control specific parts of the body. So that's like what the homunculus is. It's that these body parts that perform precise movements like hands and face are going to be disproportionately larger as far as the amount of neurons that are active when recruiting that area of the brain to perform this motor movement. So there's also contralateral processing, which I talked about with the visual center, where like things cross over. So whatever's happening on the right side of the body is being dictated by the left side of the brain, and whatever's happening on the left side of the body is being dictated by the right side of the brain. So here, if I'm moving my right hand, it's because my left side of the brain is telling me to do that. So both lobes, right, in that area that I was telling you about, the premotor cortex, it has the homunculus there. And that homunculus, as you can see, is made up of different like regions for different movements. But you can see that there are a lot more fine motor movements that are going to take place within the hands, the fingers, and the face, and the tongue. So those are the biggest ones that like have the most activity, because you got to be able to do some complex stuff with those things. So how does the subcortex contribute to movement and action? Well, it's saying that because like I was telling you, so there's the cortical regions, which is the outermost layers of the brain, the gyri, the gray matter. But then there's also subcortical layers of the brain that have some gray matter in them, not the white matter. And that's what we're going to talk about right now. So the subcortex contributes to movement and action in this way. It's responsible for the preparation and execution of action. So it's going to help to set up particular parameters, like the force and the duration of a planned or in progress movement. So force comes from one area, duration comes from another. Cerebellar loop is duration, force is basal ganglia. We'll go there in a second. So two distinct subcortical pathways for movement or the acting brain is the cerebellar loop and the basal ganglia circuit. So within the cerebellar loop, there's this idea that timing and trajectory of movement is essential. So we're talking about duration here, using sensory and motor information that uses this loop. It goes from the cortex to the cerebellum, to the thalamus, and back to the cortex. That's the cerebellar loop. So areas of the cortex talk to areas of the cerebellum talks to areas of the thalamus, and the thalamus talks back to the cortex. And that's the loop to be able to know the timing and trajectory of movement. Now, when it comes to the basal ganglia circuit, that's what's going to regulate the excitability of frontal motor structures, the SMA, and it biases the likelihood of movement and the nature of movement. So, like, how forceful should I be doing this thing? Like, um, I think about both the cerebellar loop and basal ganglia circuit when I'm thinking about like a hammer and needing to hammer a nail into a board. So, how many times do I need to hit the hammer down before the nail goes in? And am I going to be able to tell myself to stop hitting the nail into the board? Okay, that is part of the cerebellar loop. Now, when it comes to um, how hard I need to be forcing the hammer onto the nail, that is the basal ganglia circuit, where it's saying, okay, how much force do I need to be putting on the hammer to, how much force do I need to be putting onto the hammer for it to push the nail or, get the nail all the way flush against the board? That's going to be the basal ganglia circuit that's recruited. That has to do with the cortex as well. Basal ganglia and substantia nigra are both recruited. Basal ganglia, it's like the substantia nigra gives rise to basal ganglia, and the cortex also gives rise to basal ganglia. So then that ends up sending the signal to the direct accelerator or indirect brakes pathway, which so there's two different pathways of the basal ganglia, basically, that goes back to the thalamus and relays the sensory information. So um, it's saying, hey, do I need to be finely moving my body, like with fine motor movement? Or do I need to be stopping right now? Like, how fine should I be moving like in a dance, let's say, or moving your fingers across a piano versus, pushing down on something or doing a massage, you know what I mean? Like, all of those things are going to recruit the basal ganglia circuit and the cerebellar loop to be able to determine how long you should be doing that specific movement and how hard you should be doing it or how soft you should be doing it. So now when we are describing the neural basis of Parkinson's disease and its treatment, there are many details that have yet been uncovered when it comes to Parkinson's. But what we've been able to find out is that Parkinson's disease is being caused because of a disorder of the basal ganglia circuit, where, two things could be happening. There's either a hypokinetic response or a hyperkinetic response of like damage to the basal ganglia. So if it's a hypokinetic response, that's reduction in movement. And reduction in movement is called Parkinson's disease, but it's more than just reduction in movement. There's like a shuffling that takes place and a lot of neurological disorders that take place, more than just movement disorder. Hyperkinetic is an increase in movement. So Huntington's disease or Tourette's is another one with hyperkinetic issues of the basal ganglia circuit. So what's happening with hypokinetic? It's that excitatory and inhibitory neurotransmitters are showing dysregulated signals. Dopamine is not being recruited properly. So usually, like with Parkinson's, there is a mean onset of 60 years, but that's not always the case. It does affect about 15% of the population of those that are over 60. Now, the mechanism of the disease is that we have neurotransmitters and we have receptors. The dopaminergic brain cells are lost in pathways that are linking the substantia nigra and the basal ganglia. So those receptors that are supposed to help with transferring dopamine between substantia nigra and basal ganglia are lost. And in that way, now you're resulting in both motor and cognitive deficits. So why does loss of dopaminergic cells in the basal ganglia lead to Parkinson's? Well, the loops that connect the basal ganglia and the thalamus consist of two complementary routes. Remember, I was talking about the direct accelerator route, which is where there's more activity, and then the indirect route, which is the brakes kind of a route or less activity. When there is a lesion between the substantia nigra and the basal ganglia, it's going to result in an increase in the indirect pathway, which means that there's more output and a decrease in the direct pathway output. There's more output for the indirect pathway, which puts more brakes. So you're shuffling with your movement. Instead of it being fluid movement, it's like jerky. And tremors, not fluid movement. So more braking, less accelerating of the movement itself. That's resulting in a decreased ability to initiate your movements voluntarily. So how can we treat Parkinson's? Well, they found a few different ways to treat it. There's one type of a medication or a couple of different types of medications now that mimic or increase the dopamine signal. So they're called dopamine agonists. And those help basically restore the amount of dopamine needed for fluid movement. And then deep brain stimulation is another way where you can actually directly stimulate with electrical pulses, very small electrical pulses of the basal ganglia circuit to activate the direct pathway. And that is through an implanted device. But that is another way that you can do it. Okay, so I said that the acting brain was the last one. I was wrong. There is the emotional brain too. Do I want to go through this? Might as well. All right, the emotional brain. So the emotional brain. Well, there have been two historical views of what gives rise to emotion in the human mind. Now, what is emotion? Well, if you look up emotion psychologically, they're saying that it's a state associated with a stimuli or a stimulus that's rewarding or punishing, and that often has a value for survival. It's effective versus non-effective stimuli. So we use emotions through our own experiences. The mindset of others and by sharing them with others because in my opinion, emotions are like a language of the soul and they're universal. We can really speak to one another through the characteristics of our emotions, but not everyone is responsive or emotionally mature, so they can't always show up in that way. So we use the emotions through our own experiences. And here's the characteristics. a state we would like to obtain or avoid, which is like this versus punishment type of characteristic of emotion. Um, Sometimes emotions can be transient, but they're also typically stored in long-term memory. Uh, Emotional stimuli draw attention to themselves, and they have this hedonic value. In other words, there's a dichotomy between like versus dislike. You want to be in a place of things that promote pleasure for you, always pleasure seeking. An internal bodily response is like the emotional response, right? Sweating, increased heart rate. But the external emotional response is like smiling, or when you see somebody, or when you get nervous around someone and your muscles tense, or when you start to kind of shake because your nerves are shot, because you're like feeling this certain way about someone or something. That's all an external motor outcome from emotions. So early on, Darwin had a view of emotions in the late 1800s that said, nah, it's he's very much a nativist in this way, where he's saying, um, it's all about expression. There's this external motor outcome in the face and body that's associated with emotional states. He believed that expressions were innate and that they were conserved through evolution, that they were not something that was learned through experience in the real world, that it was something that just came innately. So that's a very much a nativist approach, right? Now, Freud came along in the 1920s, and his model of emotion was a bit different because he said that our minds are divided into three different mechanisms of emotion: the id, the ego, and the superego. Id being that primitive urge and basic instinctual drive, where ego is like the main decision maker between the two or the three, balancing id and superego, where superego is like this ideal based on cultural norms, or like, you shouldn't be doing that, where it is like, let's just eat that donut. Superego is like, no, you shouldn't have that because this is going to happen if you do. So that's the idea between Freud's view of emotions, is that there's um, subdivisions in our mind that make up the mechanisms for emotion. He's saying that they come from unconsciously biased behaviors, like confirmation bias, biased beliefs, usually feed into the id, the ego, and the superego that give rise to the mechanisms of emotion. And so you would say that that is learned behavior from environmental cues, and that would be an empiricist's view. So what is the relationship between neurobiology and the two theories of emotion? Well, there are different theories, and those theories led to different experiments, right? So James Lang came along and he said that emotions are mediated by the peripheral nervous system. He said that you perceive from the peripheral nervous system some sort of a stimulus, and that emotional bodily state with external or internal bodily changes will give rise to your emotional perception or interpretation. So, like, he says that the conscious self-perception of bodily changes produce your emotional experience, that changes in bodily state occur before the emotional experience. Here's an example of that. If I said, I'm sad due to the physical response of me crying, that would be James Lang theory, saying that the peripheral nervous system mediates an emotional response uh, of perception. So, in other words, like, your physical body is going to respond before your mental body does about some sort of an emotion. He said, you know, Like if I was to say my heart rate increased and I began sweating, now I'm scared versus I'm scared, my heart rate, therefore my heart rate increased and I began to sweat, right? That's the other take. How neurobiology mediates emotional responses. So that was the question, right? What is the relationship between neurobiology and the two theories of emotion? Well, the first theory, the James Lange theory, that it's mediated by peripheral nervous system, is saying that the involuntary part of the peripheral nervous system is what is triggering the perception of emotion. But there's been contrary evidence to that because they injected adrenaline into mice to see if that would produce the emotional response, right? That was not the case. The rats did not perceive fear because of having adrenaline injected into them. So they were able to say, okay, it's not that the physical peripheral response of the body is leading to the experience of the emotion. It's that the experience of the emotion is leading to the physical response of the body. And that's what Cannon-Bard found out. So Cannon-Bard theory is saying, no, actually, emotions are mediated by the hypothalamus, saying that the conscious experience of emotion is experienced and then the body responds. So you could think about that and you can put that together in a way where you conceptualize that and say, okay, well, have I ever experienced an emotion where all of a sudden, like, I realized that I was sweating and my heart rate was pounding, and then I realized I was scared? Have you ever had an experience like that? Or have you ever had an experience where instead of it being like that, it was like, oh, I'm freaked out right now. I don't know what I'm going to do. And because you keep thinking about that thing and feeling that way, now all of a sudden you're starting to sweat and your heart starts to pound. So that's what Cannon-Bard is saying. He's saying that the change in the bodily state occurs after the emotional experience. Like I'm experiencing sadness, so I started to cry. Or I got scared, so now my heart rate is increased and I'm sweating. So how does neurobiology mediate an emotional response? In the Cannon-Bard theory, he's saying that uh, with the inspiration from neurobiology practices, that the hypothalamus is the centerpiece of emotions. Animals still exhibit emotional expression after the removal of the cortex. So that means that it's not about some part of the cortex that's doing the job. The emotional expression is coming from the subcortical regions of the brain, which is the hypothalamus in the diencephalon. So the hypothalamus receives that and evaluates sensory inputs based on emotional content. And it communicates to the autonomic nervous system and the primary motor cortex. And that's what gives rise to movement or a response, emotional response rather, in the periphery. So now the Papez circuit and the limbic system. This theory came in the 1930s, where they were talking about the different areas of the limbic system. Which parts, which subcortical regions make up the limbic system? Well, the limbic system is the mammalian brain, and the mammalian brain is the emotional brain. So it's made up of the cingulate gyrus, the hippocampus, the hypothalamus, and the anterior nucleus of the thalamus. So Papez said that the emotional brain is a part of the Papez circuit plus the amygdala plus the orbital frontal cortex. Just so you know, it is no longer considered totally accurate. There's evidence now that we know that the hippocampus is much more task general than task specific, in that it is a part of the limbic system involved in memory retrieval as well. So we know that, you know, the way that this was situated before and saying, yeah, this is all just a part of the emotional brain, we know now that that is not true, that it's also a part of the learning brain, as well as the um, memory, forming and consolidating long-term memory. So, next question, I think it's almost the last one. Explain the contemporary understanding of basic emotions and constructed emotions. So, this idea of a contemporary, a newer, modern understanding of how basic emotions are formed and how we construct emotions. How are we acting on emotions? So, we all have an innate set of basic emotions, says Ekman in 1992. So those basic set of emotions, think about like the movie Inside Out, right? And you think about happiness or joy, right? And sadness and disgust and anger and fear, right? Those were the five, I think, that were like the prominent in that movie. But there's a sixth one, which is surprise. So Ekman, coined these six basic emotions that occur universally in humans automatically, saying that they are an innate type of a basic emotion that all humans experience. And he believed that the six emotions were independent of culture, that they had their own neural basis and evolutionary purpose, that they didn't come from learned behaviors. So would you say that that is a nativist approach or an empiricist approach? Is that something that comes without learning from the outside environment, or is it something that comes from uh, a biochemical response in the body? So he's saying that it's a very nativist point of view of a biochemical innate response of these six basic emotions that everyone has from birth. He says that all emotions are constructed, which doesn't make sense because they require information outside of the emotional. Okay, that makes more sense. So Bartlett in 2006, so little over 10 years later, Bartlett et al., meaning and others, said, no, emotions are constructed. They are not innate. You don't just have six set emotions that are basic that you work with from born. No, they're constructed. They come from the environment, that all emotions tap into a core affect organized across two dimensions, the idea of bodily feelings and the idea of facial expressions. So you can like you can pick up on people's emotions just based on their expressions. You can pick up on people's emotions based on their body language. That is what Bartlett et al. Was able to see, and he was able to say, no, they're actually constructed because we're influenced by our environment, right? If we see somebody's facial expression or even the prosody, right? Their pitch of the tone of their voices and the way that it switches and their vibrations and the whoops and the woos and the ha, you know that someone is upset if they're talking like this versus if they're talking like this, right? So in that way, that's going to influence the way that you are emoting. So in that way, emotions are constructed. That's very much of an empiricist's point of view. So Bartlett et al., empiricist, where Ekman was more nativist. All right. So we have more to talk about. It's a very small amount. So what are the brain regions involved in the emotional brain network and what are their responsibilities? Okay. So I'm sure everyone has heard of the amygdala. The amygdala is a very well-known brain region for emotions, but it's more than just the fear response or the primary threat center that is activated. Okay. It's actually, so it's a medial structure. It's in the medial temporal lobe, at the tip of the left and right temporal lobes, anterior to the hippocampus. So it's like if the hippocampus is this little tiny area right underneath the thalamus, right in front of that, like a little bulb, is the amygdala. So it's almond shaped, part of the limbic system, known for a basic fear response, but also fear conditioning. So we have evidence that the amygdala is also responsible for learning, for being the master coordinator in memory of specifically threatening responses and experiences. So what does that mean? That means that like if you have some sort of an experience in your life over several times, right? It's a repeated experience, then you learn the behavior of it being pleasant or unpleasant, right? So it's either going to be a threatening type of a experience or a non-threatening type of an experience. That is a type of learned behavior that you're either going, excuse me, that you're going to like or you're not going to like from someone else or from yourself. But all of that is forming within you a consolidated long-term memory. And so in that way, it's actually making up a part of who you are. So if you're losing the amygdala, you're losing the ability to develop emotions properly. So the the basolateral amygdala, the BLA, contains two populations of neurons that are genetically programmed to encode either fearful or happy memories. Why? Because what is the opposite of fear, right? Love. And what is love? Love is happiness. And so, of course, in order to experience one thing, you have to experience the other first. So, in a way, the amygdala is there for discriminating against good and bad feelings and is able to help us form and consolidate the memories around those experiences. So the anterior neurons were programmed for happiness and showed conditioning to female stimuli. In other words, when rats were tested, they were able to see that when they put a female in front of them, that was the rewarding stimuli, and that gave them that feeling of happiness. Where the posterior neurons, they were able to see that they were programmed for fear. Why? Because when the rats were shocked, they avoided that. And that area of their brain was lit up when they were shocked, trying to avoid that pain. So, legions of the amygdala in monkeys were able to give us even more evidence about what the amygdala is responsible for in emotions, and showing that the bilateral amygdala and temporal lesions in monkeys caused what's called Kluver-Bucy syndrome, which is like this unusual amount of tameness, but on a level of emotionally blunting the poor monkeys. So, like, they put a snake in front of the monkey, and instead of the monkey being afraid of the snake, the monkey approached the snake and was trying to like grab for the snake. And it had like no desire to want to eat, to want to sleep, to want to play, to want to do anything. It was like wanting to just kind of examine things with its mouth instead. You know, there was just no value of emotions anymore when the amygdala was removed. So that was really interesting. Now, the last thing about the amygdala is that it coordinates the response to fear and fear-related events. It's going to assist with learning and long-term memory storage when it comes to emotional long-term memory storage, and then it's going to evaluate the value of stimuli to make emotional associations. When it comes to the anterior cingulate cortex, which is another subcortical region of the emotional brain, it's located in the medial prefrontal cortex. There's these social and emotional functions that are activated, or whenever you have some sort of a social and emotional response to like talking and interacting with somebody or having empathy towards somebody, or like they're telling you a story and you're like, oh my gosh, really? The anterior cingulate cortex is really active. So there's like this response of evaluating somebody's story, and you're assessing like the value of the response of somebody else's, including yourself. You're determining if an action is good or bad, if it's a reward or a punishment. So um, you can see that even there's a response to pain, like in yourself pain, but also in others' pain. So if you see somebody in pain, if you see somebody like in physical pain or emotional pain, you respond And that part of your brain is active to promote empathy. So if you have a lesion in your anterior cingulate cortex, a lot of the times it's shown that you lack empathy, that you're very apathetic in that way. And it might just mean that you don't have good white matter tracts in the anterior cingulate cortex that allows for that type of a process to take place. But um, yeah, so receives pain sensory input from the thalamus. It regulates the feelings of pain. This is all ACC or anterior cingulate cortex. It connects to areas rich in endogenous opioids, which is also really important. This is something that Candace Pert found, the opioid receptor. Yeah, and then the insula is like a small, insula means island, and it's this small region that's actually a subcortical region. So it's actually a cortex within the subcortex that is specifically there for interoception. So whenever you're feeling what's going on in my body? How am I feeling? Is my back hurting right now? Like my lower back really needs to be stretched. That kind of thing. Um, Feeling your heartbeat, feeling your temperature internally, all of those, that's all like something that's being recruited from the insula. And also the feeling of disgust. So they actually did a study where they showed pictures of people that looked disgusted. And that area of the brain was activated when people were looking at other people disgusted. Very interesting. So, whenever you like experience a actual physical bad taste, that area of your brain is going to be lit up. Moral disgust. If you're disgusted with something morally and you're like, no way, are you kidding that happened? Ew, clench my pearls. That is insula. The orbital frontal cortex is a really neat area located in the prefrontal cortex that computes the current value of a stimulus within a current context. So it's like, should I respond or should I not respond? That's actually recruited by the executive function of the frontal lobe, right? Contextualizes emotions. It links to subjective reports of pleasantness. Um, and it's going to help you quickly change your behavior to one thing or another, discriminating against what's going to be pleasant or what's not going to be pleasant. How am I wanting to make a decision? Do I want to say yes to this thing or no to this thing based on how it's going to make me feel? Lastly, and this is it, is the ventral striatum, which is located in the basal ganglia being the subcortical region of the brain. So the basal ganglia is going to be found close to the diencephalon, and it connects the orbital frontal cortex and it specializes in your emotions by calculating the probability of a reward in social and non-social contexts. So it's going to connect the frontal cortex um, to increase or decrease the probability of reward. So that is very much connected with the orbital frontal cortex in that way, right? That's why it's saying that it's located in the basal ganglia and connects to the orbital frontal cortex, because both of them are involved in trying to discriminate and determine how you're going to make your decision based on whether it is a good outcome or a bad outcome with good probability. Okay, so that is basically it. I really hope that this helped you study. It definitely helped me study by reiterating all of these aspects of cognitive neuroscience. And again, just to kind of recap on what we went over in this um, session was all about the seeing brain and the visual pathways of the geniculus striate and learning about the different regions of the brain that get lit up for visioning, as well as the auditory brain, the hearing brain. We went over the acting brain, and we went over the emotional brain. So I hope that helped. And again, I will have another video coming up. And yeah, best wishes for an amazing finals week. I know we're all going to do great and lead on to even bigger and better things to help humanity be bigger and better people. Take care. Bye for now.